Sorry to dominate the conversation here, Brendan. I apologize. I don't mean to do that. But
Absolutely not. You're asking great questions. And you're so much more articulate than I am. I'm glad you're doing it.
not on my best day. Are you kidding me?
Uh,
This guy.
so, uh, so I'm going to ask a question. I have to edit out because it might be so stupid, but I'm wondering about it. So you mentioned this virus in particular seeking and finding certain types of cells. Um, so do I have it right that each virus has a certain kind of cell that it attacks? Um, for example, HIV attacks immune cells. And I don't know the medical part of exactly what kinds of cells are attacked by this virus. If you can talk about that, fine. If not, maybe that's too much on the medical side. But is there a certain type of cell in an animal or a human that is more vulnerable to um, an attack by a virus than other types of cells?
That's a good question. Yeah, so viruses infect very specific kinds of cells. In general, they're adapted to infect those particular types of cells. And so this virus infects cells that are in the respiratory tract. So it's a respiratory infection. That makes sense. The way that it recognizes what cell to infect is that it has that spike protein. That spike protein has an architecture and has chemical properties on the outside of that protein so that it senses a protein on the outer membrane of human cells and bat cells and so forth. And that protein is called ACE2. And so this virus is good at binding ACE2 of humans. So if you just sort of think back in biology, all organisms are descended from a single ancestor 4 billion years ago, a bacteria. You know, bats and humans shared a common ancestor much more recently. And so a lot of bats have very similar proteins as humans have. They have similar proteins on their outer membrane as humans have on their outer membranes. And so do chimpanzees, and so do, you know, all of these different mammals. And so a virus that is able to use and recognize that ACE2-like protein in bats could mutate in a way that it could begin to use ACE2 proteins in humans. And so that's what's happened, is that it was using a similar protein in bats to recognize those bat cells and to infect those bat cells, and some kind of genetic variation accumulated in those proteins that allowed them to now recognize human ACE2. And so the way that you can think about this interaction is it's a lock and key interaction. You have that spike protein, which is the key that unlocks that ACE2 protein in humans. And so normally with that metaphor, people have thought of these interactions as being very specific. You have one lock and one key. But what we're learning more and more is that you can have a skeleton key. You can have these host recognition proteins, the spike protein, that can recognize lots and lots of different locks. And so it appears that this is one of those types of spike proteins that can recognize lots and lots of different ACE2 proteins in humans or in bats or in pangolins or wherever. And so, yeah, so there is this, you were kind of connecting that there's sort of specificity of viruses. They infect specific types of cells in an animal's body, but they also have specificity in the types of species that they infect. And so somehow these specificities of tissue type and cell type and species play out so that most of the time viruses are constrained to a very particular type of tissue and species, but there are characteristics that allow some other viruses to jump. And so I was mentioning EcoHealth Alliance, and one of their big questions are what are the characteristics of viruses that allow them to be more promiscuous, allow them to have these skeleton keys. That's something that my lab works on as well in different viruses. And we're sort of at the very beginning of understanding what characteristics produce that. The other question that you had was about, are there certain cells in the body that are more vulnerable to being infected and exploited by viruses? And I would say yes, that's certainly true. I haven't thought about this in great detail, but certainly any cell that is external and basically a virus can get to more easily than other cells. Obviously, that's going to be one that's more vulnerable to being infected. And so a virus that specializes in, cells that line your lungs, where they're always getting exposed to, particles coming from outside the body, inside the body, that's going to be a vulnerability. STDs are a vulnerability because, those are viruses that exploit cells that humans interact with each other with, and so that's another vulnerability. There's lots of them. I don't have an exhaustive list, but there are characteristics of cells that make them more vulnerable.
I have to interject here and take strong exception to something you said just a minute ago. You mentioned all life having evolved over the past 4 billion years. Well, I have it on authoritative and reliable information that life has only been on this earth and indeed in the universe for 6,000 years, mister. You are blaspheming our Lord and Savior, Jesus H. Christ, and I won't stand it.
Yeah, I mean, the Catholic Church and most churches have believed in evolution for a very long time, and there appears to be no conflict. It's just a particular branch of zealots in America that push that idea that science and religion are incompatible. I know a lot of evolutionary biologists that study evolutionary biology because they think of it as the algorithm that the creator uses in order to produce creation, in order to produce all this biodiversity. So there isn't an inherent conflict. There's a YouTube video trashing my research specifically. There's
no kidding.
A lot of air quotes around mutation.
Congratulations! That's how you know you've made it, right? Yeah. If you've got the creationists coming after you, you know you must be doing something right.
Exactly.
Yeah. You have arrived.
And you especially got them angry. I was actually going to ask you this earlier, so I'll ask you now. Because, and again, I have such a limited understanding of this, but it's not just that you study evolution, but also speciation, right? Is that part?
Oh, that pisses them off. Oh, that pisses them off.
There's two subjects that we have so what makes my research special is that I don't sort of just watch viruses evolve in nature. I culture them in the lab, and so I can reconstruct how they evolve under these different lab conditions. And I set up unique scenarios for the viruses to adapt to and to change to. So I try to study, just normally how virus adaptations work. But my real niche in all of this is that I want to go after these larger questions in evolutionary biology. And so one of the big questions is, how do you evolve new functions? So if you think about, how natural selection works, it's always about improving at the things that you're already doing. Well, how do you take that step to do something brand new? That's why my research relates to SARS-CoV-2 jumping into humans. It's doing something brand new. So the theory for how that transition, how these big steps in evolution happen, is a little flimsy, and we don't really understand those processes. And so when I was in my PhD, I set up an experiment where I wanted to evolve my virus to use a new receptor. We know receptor usage is important for these host transitions, and it's a case where it would be doing something new, and it would be changing its host range, changing its ecological niche. And so we sort of check all these boxes as being an innovation. And then if I saw that happen in the lab, then I could reconstruct exactly how it happened. And so that YouTube video that I talked about is trashing that research, because a big a line that the intelligent design people have made in the sand is that organisms might be able to adapt through microevolution,
Right.
that they can't do these larger things. And so one really arbitrary line in the sand was that this was by a professor in Pennsylvania, Beery, and he said that, well, okay, you can get microevolution, but if an innovation required more than 3 mutations, that would be impossible. Well, our virus evolved to use a new receptor, and it requires 4 mutations, so it broke through that barrier. And now I've spent a lot of time trying to understand how exactly it evolved this innovation. We have a lot of papers on that. So that's sort of one section of research that they hate. The other thing, after I observed that in my PhD, I thought, well, if I have a virus now that can do 2 things, use this receptor and use this other receptor, that then sets up a scenario where you could get the virus evolving in one direction or the other direction. And anytime you have natural selection that can now split things into 2 unique ecological, 2 unique viruses that use different receptors, use different host cells. These establish 2 different ecological niches. And if you play that selection out long enough, you can actually, or at least theory predicts that you should observe speciation, where these viruses or other, this is happening all the time in other organisms as well, become so distinct that they become separate species. And viruses evolve fast enough that you could actually see this on the timescale of laboratory experiments, a couple month-long experiments. And we observed this with my virus, that you would get completely unique viruses that were no longer able to recombine their DNA with each other, which is the sign that these are unique viruses that are independent species. So we showed speciation in a laboratory setting, in a controlled setting. We were able to reconstruct all of the mutations that contributed to their evolution and their speciation. And so that's another big bar for intelligent design, is that, okay, well, you can get the set number of species that came off the ark, improving, but you don't get the origination of new species, where we've shown that now with laboratory experiments. So when you can, a lot of the doubt for evolutionary biology is that it takes so long where these processes unfold over so much time that it's impossible to observe. But my PhD professor and other professors in the past realized that you could watch these processes actually underway in microbes under controlled settings and within a reasonable amount of time, so you can actually see evolution happening in action. So it's really changed the game in understanding evolutionary processes, but also, proving intelligent design people and their assertions wrong.
Yeah, so you're a dangerous person.
Yes, you are. You are the devil incarnate, my friend.
I know. I always think like I'm a gay evolutionary biologist.
Where's the mark of the beast? You must have the 666 somewhere. That's all I can say. Where
Where did my parents go wrong?